Good afternoon and welcome. I'm Ellen Siegel, Chair of Friends of Cancer Research, and welcome to our third virtual meeting. I would like to thank all of you and your incredible partners on this important project and welcome you to Friends of Cancer Research, a meeting on ctDNA. Friends launched the ctMoniTR project in November 2019. The truly impressive collaborative research project was brought together by the best and the brightest from pharmaceutical companies, diagnostic labs, government health officials, patients, and academic researchers. We are thrilled to be able to share new data from this endeavor today and have an impactful discussion about the future of ctDNA. And of course, the most vital is how this impacts patients. I am pleased to turn this over to Jeff Allen, CEO and President of Friends of Cancer Research. And again, a giant thank you to all of our collaborators and all of you for your hard work on this project.
Thanks so much, Ellen. Good afternoon. I'm Jeff Allen, President and CEO of Friends of Cancer Research. Thanks very much for joining us today for the initial presentation of Friends ctMoniTR Step 1 results. This research partnership was launched to begin to answer the question, do changes in circulating tumor DNA reflect response to treatment? And we're excited to share the results today. ctMoniTR is the outgrowth of a Friends of Cancer Research annual meeting. Like many of our projects, we convened a multi-stakeholder working group to begin to address a current challenge to developing new therapies. In this case, the work group proposed a uniform method for collecting and evaluating ctDNA that could be embedded into individual clinical trials. The idea was to create a synergistic effect, where the results of the partnership can be even more informative together than as individual parts. We're pleased to have the opportunity today to hear from several leaders involved in the discovery, development, regulation, and application of ctDNA, as well as an overview of the results of Step 1, which is the first of 2 steps in the ctMoniTR project. I want to thank all of the collaborators that have participated in this work, sharing their data, expertise, and time in order to determine a stepwise manner if levels of tumor DNA found in the bloodstream can be an accurate predictor of whether a drug is working. To start that project's results discussion and to set the stage, I want to introduce Dr. Julia Beaver, Chief of Medical Oncology, the Oncology Center of Excellence at the US FDA.
Thanks so much. I'm very excited to be able to participate in this impressive meeting. And a huge thanks to Ellen Siegel, Jeff Allen, Mark Stewart, and Diana Marino for spearheading this work. Friends of Cancer Research has yet again delivered on their promise to drive collaboration and power advances in science, policy, and regulation with the ultimate goal of speeding life-saving treatments to patients. This project was initially conceived through discussions surrounding a prior Friends of Cancer Research annual meeting, where a group of experts discussed the role of ctDNA for monitoring a patient's tumor response, discussing evidence related to this, and concerns that the evidence came mainly from a disparate group of smaller studies with different collection methods, criteria, and retrospective data analysis, making the broad learning or conclusion generation difficult. Through feedback from this meeting and further work with many collaborators, the framework for this project was created to fill the gaps and initially assess if through a structured evaluation, we could still demonstrate across multiple studies within a single tumor and treatment type that ctDNA reflects response to treatment, exploring methods to account for differences in the various study designs. Now, at the completion of Step 1 of this project, several important points have been demonstrated. 1, it is possible to bring together different industry and academic groups to share patient-level data on ctDNA studies. Second, it's possible to harmonize and gain knowledge using a heterogeneous grouping of trials despite different assays with different collection schedules, different levels of clinical detail, and different raw ctDNA data value points. Third, the association between ctDNA values and tumor response remained from trends observed in the individual small trials into the structured combined data set, which is very exciting. And lastly, this has really set a strong foundation for future directions to work on guidelines for standardized ctDNA assessments or quantification, and adding to the body of evidence to support the fact that ctDNA does reflect response to treatment. Of course, the question everyone wants answered relates to how ctDNA could be used as an efficacy response biomarker or putative surrogate endpoint in drug development, and what future studies or efforts would be required to allow for that use. And we all realize the promise of ctDNA in that if ctDNA could be shown to predict benefit of a drug at an earlier time point, it would allow for drug development to be expedited and drugs to receive earlier approvals compared to waiting for progression or relapse or survival. Important considerations related to this include demonstrating that ctDNA is not only prognostic, but can predict treatment effects in the metastatic solid tumor setting, and demonstration that ctDNA reflects long-term outcome in an improved or complementary way to tumor imaging. And in the early stage setting, there are opportunities as well related to identification of high-risk disease and with answering questions regarding association with earlier ctDNA response and long-term outcome. We're excited to discuss and review your ideas, and I look forward to hearing the discussions today and to continuing the dialogue with friends in step 2 of the collaboration going forward. The NMD group, Cancer Research and Biostatistics, and all the collaborators involved should be extremely proud of your efforts. This project was rapidly brought forward and the ambitious timeline was maintained. This is the exact type of effort that is so critical if we plan to advance the field of ctDNA, understand and standardize the use of ctDNA, and gather evidence to further support the ability of ctDNA to inform, if not drive, drug development. Thank you, and I will turn it back over to Jeff.
Great, thanks so much, Julia. Just to note, Julia will be available for questions following our next discussion. And in order to submit questions for her or any of the panelists, please use the question and answer function on the bottom of your toolbar. I'd like to now turn to the anticipated results presentation for today's meeting. Dr. Diana Marina Vega is the Director of Research Partnerships at Friends of Cancer Research. She's been the project lead for ctMoniTR over the last several months and has expertly guided a growing work group of advisors and participants to create the ctMoniTR project. Diana, thank you so much for your incredible work. I'm pleased to turn the presentation over to you.
Thank you, Jeff. My name is Diana Marino Vega, and I'm Director of Research Partnerships at Friends of Cancer Research. It's an honor to present the ctMoniTR project results on behalf of the ctMoniTR Working Group. As you can see, the ctMoniTR project is a team effort, and I would like to acknowledge our numerous partners without whom this project would not be possible. I would like to especially recognize the work and talent of the Cancer Research and Biostatistics team led by Dr. Antje Hoering, especially Dr. Katie Nishimura, the Senior Biostatistician for this project, Vanessa Salento, and Adam Rosenthal, as well as Matt Scott. Additionally, a special mention to Nevine Zariffa from the NMD group for her expertise. As Jeff and Julia previously mentioned, the ctMoniTR project consists of 2 steps. Step 1 will set the foundation for future work. We were primarily interested in determining whether trends observed in independent data sets could be replicated in aggregate. To do this, we had to address several challenges, such as developing tools for streamlining the data intake processes, understanding the existing ctDNA data available, evaluating the feasibility of harmonizing ctDNA data generated by different sources and harmonize the clinical data, and analyzing the relationship between ctDNA values and tumor response. You will see in today's presentation that we have achieved our goal. We now ask ourselves, what are the research questions we should prioritize for step 2? And what can we learn from step 1 to develop an informed framework for step 2? This project has generated results very quickly, and this is largely due to the invaluable support of our partners. Not only did they commit data, but they also provided their time and expertise at every step of the journey. The statistical experts met several times a month, and all partners met in January and May to finalize the statistical analysis plan and review the preliminary results respectively. Here you can see the steps we took and the pace at which we progressed. I will start by describing some of the challenges we faced with the disparate data sets we received and how we were able to find solutions for data harmonization. The ctMoniTR working group decided on a set of criteria that would ensure success while balancing scientific robustness within a rapid time frame. The criteria used for the collection of data included immediately available data from patients with advanced non-small cell lung cancer who were treated with an immune checkpoint inhibitor, and we required studies to have evaluated tumor response, collected survival data, and a minimum of 2 ctDNA measurements, 1 at baseline and 1 or more follow-up samples, assessed and reported as variant level data in the form of variant allele frequency, or VAF. The chart on the right shows the sample size of the pooled dataset used for the final analyses, which included data from 5 sponsors split into 7 cohorts with a total of 200 patients. Datasets used in this analysis were originally designed to examine different research questions, so they used different collection time points and had their own eligibility criteria. Therefore, we expected and observed many differences across datasets that could provide biased results when analyzing pooled data. There were 5 potential sources of bias that we highlight here. The retrospective nature of the datasets meant that we could not retrieve additional data elements or request a new sampling schedule for consistent data collection across all cohorts. We had to use the data we had. We discussed various solutions with our study partners to determine optimal approaches that would mitigate any downstream bias introduced by these features, while prioritizing inclusion to maximize the number of samples. This was done prior to performing any of our analyses. We believe that the approaches used to address each problem greatly reduce the noise and bias inherent to these data. On the next few slides, I will describe some of these solutions. Inconsistencies in the timing of ctDNA assay and tumor response evaluations across the datasets were apparent very early on, as this figure clearly shows. Every row represents a patient, and these are ordered by follow-up time. The pink dots represent the timing of the ctDNA sampling, and the blue dots represent the tumor response assessments. As you can see, the number of ctDNA samples per patient also differed by sponsor. We used timing windows to ensure that samples that were overly dissimilar from the rest of the data were excluded. We also conducted a sensitivity analysis to determine the influence of ctDNA timing, but we didn't have the right type of data to draw any significant conclusions. Another challenge was the variability in the clinical detail provided and heterogeneity of patient populations in the study. We evaluated each baseline clinical trait, most of which we collapsed into binary and categorical variables here outlined in red. In the blue outline, you can see imbalances in the clinical covariates across cohorts, which provided rationale for the importance of stratifying the statistical models by cohort. The rows in bold are clinical traits that differ by cohort and were thus identified as potential prognostic factors in downstream analyses. As a result, we chose to include all clinical traits in our statistical analysis plan. I will point out that some of them, such as smoking and PD-L1 expression, remain statistically significant in multivariate models, though those findings were inconsistent. Many different methods are currently being used to quantify changes in ctDNA. Thus, we developed and evaluated several approaches. Obtaining variant level data from each patient was key in our ability to explore different types of ctDNA metrics. Because of the data available, we used 2 ctDNA time points for all samples. Baseline ctDNA, which included plasma collected no more than 14 days prior to start of treatment, and time point 1, or T1, a collection no later than 70 days from the start of treatment. The mean, median, and maximum VAF were calculated for each ctDNA sample, and then the percent change in the VAF values from baseline to T1 were calculated. The continuous percent change variable, in the blue box, was the percent change of ctDNA from baseline to T1. patients with a negative percent change value had a decrease in ctDNA from baseline, and those with positive percent change an increase. The ctDNA metric proved to be the most difficult to analyze because of the presence of a few large genuine outliers. Even after the outliers were capped at 500%, in general, the ctDNA metric was the weakest of the three. The two-level variable in the green box was defined after performing an optimal cut point analysis, which identified negative 50%, a 50% decrease in ctDNA, as an optimal cut point that maximized differences in overall survival. Patients were assigned to two groups, having a large decrease in ctDNA that exceeded 50% or having an increase or minor decrease in ctDNA. The three-level variable in the yellow box was created to investigate trends in ctDNA changes, as well as take into account cohort-specific differences in the ctDNA results. Within each cohort, patients were ordered based on their ctDNA percent change values. Among patients with decreases in ctDNA, the 50% with the most extreme decreases were assigned to a decrease category. Among patients with an increase or no change in ctDNA, the 50% with the most extreme increase were assigned to an increase category. All remaining patients were assigned to the intermediate category. All three of these types of ctDNA metrics were calculated for mean, median, and max VAF. However, for simplicity purposes, we will only show results for max VAF, which had the strongest and most consistent signal across multiple analyses. 4 clinical outcomes were assessed in step 1. Overall and progression-free survival were evaluated using Cox proportional hazards models. And logistic regression models were used to evaluate partial response or better, or PFS, more than 6 months. In addition to the ctDNA metrics, we also evaluated common clinical descriptors as well as baseline ctDNA values through univariate, bidirectional, and full multivariate models to adjust for confounding by key factors. All models were stratified by cohort to account for cohort-specific effects. Now we will move on to some of the highlights from the project results. We observed strong associations with overall survival, here on the left, and progression-free survival on the right for the 3-level ctDNA metric. The strength of association across levels was quite pronounced with a clear gradient. Patients who had a strong decrease in max VAF, the dark blue curve, were associated with the best survival. Patients who had a strong increase in ctDNA, the red curve, having the worst survival, and patients in the intermediate category, the green curve, being somewhere in the middle in between. All but one of the pairwise p-values comparing the individual curves were significant. Similarly, strong findings were seen in the 2-level ctDNA metric, where patients who had a strong decrease in ctDNA had better OS and PFS. Multivariate Cox models demonstrated that the 3-level ctDNA metric remained strongly associated with OS, as shown in the figure on the left, and PFS, shown in the right, even after adjustment by other key clinical covariates. The orange box in the last row of these figures highlights the hazard ratio greater than 1, which indicate increased risk of an event, in this case 2.28 times the risk for death on the left and 1.76 times the risk for disease progression or death on the right, with each increasing level of the 3-level ctDNA metric, so from strong decrease to intermediate to strong increase groups. Finally, we also observed that strong decrease in ctDNA is also associated with improved patient response, defined as partial response or better based on the 2-level VAF percent change group. The downward sloping dashed lines for each cohort show that the predicted probability of achieving a partial response or better declines in patients who had an increase or weak decrease in ctDNA. Logistic regression analysis of the aggregate dataset revealed an odds ratio less than 1, in this case 0.12, demonstrating a strong reduction in the odds of achieving partial response or better in patients who had an increase or weak decrease in ctDNA. So, as you have seen, step 1 of this project achieved all of its milestones and produced some very promising results. Through careful and strategic harmonization strategies, we have shown that disparate ctDNA datasets can be aggregated and analyzed together. Decreases in ctDNA were found to be associated with better clinical outcomes in multiple analyses, including overall progression-free survival, durable clinical benefit, defined as progression-free survival at greater than 6 months, and tumor response, defined as partial response or better. Interestingly, we did not find evidence that baseline ctDNA values were predictive of clinical outcomes, implying that the change in ctDNA from baseline is a stronger indicator of response to treatment. Finally, these associations remained even after accounting for cohort-specific differences in other clinical covariates. We were able to advance our knowledge substantially from the retrospective data used in step 1. As a proof of concept, these data gave us the opportunity to explore and experiment with different approaches. However, there were also limitations to the type of questions that we were able to answer with these data. If we had several ctDNA samples per patient collected at regular intervals, starting early during treatment, we would be able to investigate how early do changes in ctDNA predict patient outcome, a crucial question that would allow clinicians to make faster treatment decisions. Likewise, if we had more data from multiple tumor types, disease stages, drug classes, lines of therapies, and diagnostic assays, we would be able to determine whether the association reported in step 1 is consistent across all scenarios or only significant in some. Step 2 of the ctMoniTR project provides stakeholders an unprecedented opportunity to further investigate key questions related to ctDNA and accelerate our understanding of the role of liquid biopsies in cancer research and care. So, we are asking for partners to join us for step 2 of the ctMoniTR project. A rapid and ambitious timeline, as we hope to read out sometime in 2022, will give us the advantage to drive some key conversations and propose data-driven solutions that will forge the path ahead in this innovative field. It is thanks to collaboration of this magnitude, which aggregates not only data from multiple partners, but also their vast expertise, that we will be able to expedite research and drive innovation in a way that translates to rapid patient impact. Thank you very much for your attention and interest in our work.
Great, thanks, Diane. Great presentation. And thanks so much for your outstanding leadership of this project. We have a great lineup of experts today to discuss these findings. I'd like to turn things over to our moderator for today's discussion, Dr. Nevine Zariffa, principal and founder of the NMD group, who's been an instrumental advisor and architect of ctMoniTR. Nevine?
Thank you, Jeff, and it has been a pleasure to be part of the leadership of the project. We now move on to the interactive portion of our time together. For the next half hour, I will be joined by a number of panelists. We will take your questions as they accumulate in Q&A. Allow me to introduce the panel. We're going to start with Dr. Roy Herbst, who is a professor of medicine and of pharmacology at Yale. He's also the chief of medical oncology and the associate director of the Translational Research Center. Roy.
Thank you. It's a pleasure to be here. Congratulations on the results.
Thank you and welcome. If we move on next to introduce Dr. Antje Hoering, who is the president and CEO of CRAB. This is the statistical expert team that conducted the analyses that Dr. Marino Vega has just shown us. Antje, welcome.
Thank you, Nevine. And I'm very pleased to be part of this panel, and we at CRAB are very honored to have been the Independent Analysis Center for this exciting project. I have to say I've been really impressed by Friends and how they put together such a diverse team of experts from pharmaceutical companies, diagnostic companies, academic institutions, and the FDA, and then ensure that it's a productive collaboration with high quality work and robust and exciting results. And like Diana was saying earlier, it's been done in a record time. So Dr. Diana Marino Vega and Nevine Zariffa have been instrumental in moving this project ahead, and I'm very grateful for their leadership. And our regular meetings with the statistics subcommittee have been equally important. It's been a true honor to work with this group of experts. I'm also really proud of my statistics team at CRAB with Dr. Katie Nishimura as the senior biostatistician for this project, Vanessa Silento and Adam Rosenthal, as well as Matt Scott. So it's been an intense few months, and focusing on this exciting research project has helped us actually during these challenging times. So thank you, Nevine.
Thank you, Antje. Welcome. Next on the panel is Dr. Jeff Oxnard, who was one of the leading researchers on ctDNA. He is now at the Foundation Medicine as VP and Global Medical Lead there. Jeff, welcome.
Thanks so much for having me. I'm really glad to be here on behalf of my colleagues at Foundation Medicine and Lexington Biosciences, a recent acquisition at Foundation Medicine, who contribute to the data and the development of this effort.
Thank you, and welcome once again. And to complete our panel today, Dr. David Raben, who is in Genentech. He is the VP there in the Medical Oncology Development Organization, in charge of medicines for lung cancer as well as head and neck cancer. Welcome, David.
Nevine, Jeff, Diana, Ellen, and of course, my esteemed panelists. I'm really honored to participate in this panel discussion today. And I really want to thank the Friends of Cancer Research for their coordination and the collaborations over the last year. It's really been a wonderful journey. And I also wanted to thank members of our team at Genentech who have really offered tremendous input, data, and advice to better understand this effort. So again, thank you very much for having me.
Great. And while we start to process all the questions we're getting in the Q&A pane, I want to start off with just your initial reactions. What has been most striking to you as you reviewed the results? And let me see, Jeff, why don't we start with you?
Really, it's the multivariate analysis that impresses me so much, and that the signal from ctDNA is so convincingly prognostic, even when controlling for all these other, well-established prognostic factors. And so it really makes me very hopeful about how we could start using this in the near future.
Very good, thank you. Roy, I wonder if you might add to that.
Right, I think it's absolutely amazing, exceeded the expectations, because we've known with immune therapy, it's hard to predict response, and there are so many variables. And, a few years back, Sarah Goldberg and Abhi Patel and the group at Yale did 25 specimens using our own platform and showed a very similar trend. But now with these large numbers across multiple platforms, harmonized data sets from multiple companies and institutions, I think it's just so profound that with a decrease in ctDNA, the effect we see on response, on progression-free survival and survival, this is a tool that's going to be so important to us, both in clinical care and in research for years to come. So when I was asked to join this panel and I saw these results, I was just so excited to be here today, because this really is a great advance.
Thank you so much. And David, I wonder if we might ask you to comment as well. What was most striking to you in the results?
Thank you, Nevine. Building on what Roy said and the presentation, the wonderful presentation Diana gave, we initially paused and asked, is this too good to be true? And what I mean by saying that is that despite some of the anticipated limitations mentioned in the presentation you heard just recently, we really are excited about the generated data and what this could mean. I think the message that ctDNA indeed may truly be a powerful tool to overcome the variances and the feasibility challenges really jumped out at us. And so we're really motivated, enthusiastic to continue this collaboration with Friends of Cancer and our industry partners and in academia, and hope to build on this initial experience and potentially explore ctDNA in other settings. And we understand fully that we can't do this alone. So it's a great first start and something to really build on.
Very good, thank you. And Antje, your team was closest to the data. I wonder if you might comment with your reactions.
Yes, I'm happy to. As you know, it's been a very ambitious project with a very aggressive timeline. And honestly, initially, we weren't even sure that we could harmonize the data at all from all these diverse data sets, especially the circulating tumor DNA data. So I've been truly amazed and encouraged that we could harmonize the data sets, and that using all these different assays and the pooled analysis we received very, very strong and robust results. And Diana wasn't able to show all of our analyses. We've done many additional analyses and sensitivity analyses. As she said, we did overall survival, progression-free survival, response by RECIST, progression-free survival at 6 months. And then we also looked at the different metrics for ctDNA. And finally, we also did a sensitivity analysis where we looked at different time points for the ctDNA samples after initiation for therapy. The analysis she showed was based on the ctDNA measurements for patients' earliest available post-baseline sample. We also did it for the samples taken closest to first clinical response assessment. And all analyses have always shown consistent results. Strong decrease in ctDNA within 70 days after initiation for therapy are associated with superior clinical outcome. And we found that in univariate analysis and in multivariate analysis after adjusting for known clinical prognostic factors. So it's just been amazing just how consistent it's been, giving diverse data sets.
Thank you for that. Thank you all for that. And Antje, please don't leave the hot seat. I have a question around collaboration and sharing of data. And of course, this is always a hot button issue. We had options when we first looked at this, and we're talking with our collaborators ranging from what you've seen here, which is sharing of raw data, individual patient level data, all the way to, okay, well, we'll conduct the analyses and just send you the results. Antje, as the statistician on the panel, I'd like to ask you first your reactions to that. Did we make the right decision? What were we able to do as a consequence of collaborators trusting you with the individual patient level data? And then I'm going to go to probably Jeff or David to speak some more from the non-statistical standpoint.
Yeah, thank you so much, Nevine. You know, truly, we could not have done these in-depth analysis without patient-level data. The clinical studies were designed with different objectives in mind, and the assay data from the various assay companies were different. So each data set was really unique, and we needed patient-level data to properly harmonize the data and avoid potential bias. If you get patient-level data, there's a number of benefits. There's more options for harmonizations. There are more options for statistical analyses. And there's also efficiencies to be gained rather than having single independent groups do all the data processing and the back and forth with the individual groups. Statistically, it's more powerful, so we couldn't have done it just as a meta-analysis. And I understand the sensitivity and confidentiality with regards to data sharing, and I truly appreciate that our sponsors trusted us with their data. I mean, at CRAB, we take data confidentiality very seriously, and we know how important that is. So thanks.
Very good. Thank you. David, Jeff, maybe first.
I think we should recognize how innovative this analysis is, right? We are really sort of laying down the groundwork of how to do these analyses going forward, and trying to impede sort of the degree of data that's available really will slow that process. And so if we want to enrich the most, sort of the fastest progress and the most innovation, we need the data available. And what I really admire from the work Friends of Cancer Research does is how you are able to control for all the identifiers and make sure that, we sort of blind the data from the data set behind it all, so that we can learn as much as possible while maintaining the confidentiality of what the sponsor has provided.
Very good, thank you. And Dave, from the sponsor side.
Yeah, no, just building on what I just heard here, I would agree. I think patient level data is the gold standard. But to get that data on the level needed for these types of efforts is somewhat of an operational challenge. And there are clearly some other considerations giving the industry perspective as it pertains to the timing of data sharing. But nevertheless, understanding these complexities, these efforts offer really a wonderful opportunity to build on the data sharing experience and what we've learned so far.
Great, thank you. So hopefully we'll have more of that in step 2. So I'm starting to make my way through the questions that we're receiving. And the first one, I think I want to direct to you, Roy. And it's a question about, okay, how does this step 1 data inform the next wave of clinical trials or of clinical practice?
Well, now that we have these data, it now needs to be looked at prospectively, of course. The cutoff was picked, and we saw from Antje, how that was done. But, the next step will be to do prospective trials, separate predictive from prognostic markers. But I can imagine doing a series of trials where we include this type of marker to follow response. And then, of course, we're moving in lung cancer to earlier stage disease, adjuvant therapy. I just recently presented the ADORA trial, for example, at the ASCO meeting. Now, that's not immunotherapy, but same thing, a target, and we want to follow minimal residual disease. So this type of assay is going to be huge. But right now, I think the next step will be there are so many immunotherapy combinations. And very many of these don't have single agent response rates. So if we want to see that we're having an effect, and I think that this can be used and incorporated into clinical trials, prospective randomized trials. And I, for one, will want to incorporate it into things like the lung map trial now. And we have the lung map trial going on with IO combinations throughout the country. This can be immediately added in. And fortunately, we are already collecting the samples to do it. So it's really great.
Wonderful, thank you. So we have a question around the timing and what can we elucidate in this step 1 around timing earlier in the 70-day window versus later in the 70-day window. Antje, I know I have some recollection of the work done there. I don't know if you also would like to speak to that.
Yeah, I'm happy to, Nevine. So as you, Diana showed that graph for each patient and when the samples were taken and when a patient responded, and you saw there was huge variability. And so the samples or the data we had a baseline and at least one other sample. Ideally, we would really want to know, is the timing dependent on the type of therapy? What's the best timing? Is it dependent on tumor type, on disease stage? And in order to do so, it'd be fabulous if we had more data points, especially early on in sample collections, in therapy.
Very good. So I think you've got your first vote for a question that could be answered in step 2 prospectively there. Jeff, I think you may want to add something to this point.
ctDNA is incredibly dynamic. It actually clears very quickly. The half-life in the blood is a matter of hours. And so one of the things we've seen in studying ctDNA response in EGFR mutant lung cancer and some other smaller data sets is how quickly this can change. What I mean is that within a matter of weeks, you can get dramatic clearances of ctDNA. And so that is exciting, that it has the potential to be an earlier marker than protein. Protein sort of spends more time in the blood and actually is a later signal, or imaging. We know with imaging, you can get a response on your first scan or your subsequent scan or the later scan. And so if we can really zoom in on that early opportunity to see an effect, I think that could both help patient care and could help drug development. Now, what timing is realistic, right? When does your patient come back in for their first follow-up visit? In my practice, that tends to be, a couple of weeks after therapy, they come back in for a toxicity assessment. I think that 2-week time point is really a great space to look at, maybe 4 weeks, but certainly before I think you get your first scans would be ideal going forward.
Very good. So I think we're now getting even more specificity around this question for step 2. So let me carry on just for a few minutes with questions that have to do with the data analysis. So all of the audience can be on the same information base. So there's a question about the cutoff that we chose, the 50% cutoff as an evidence of strong decrease. That was independent of the assay's limit of quantitation, which is typically much, much lower actually. So just to answer that one, there's also a question on whether or not we were able to look at any other treatment classes other than PD-1 monotherapy, so combination therapy, and that was not part of the retrospective data set we had for step 1. So just maybe going to use this as a launching point to the panelists. In terms of step 2, where would you prioritize our energy in terms of drug classes? Let's see, who might want to start? Dave. Yeah, Dave, if you would start with that, that'd be great.
Yeah, I think that's a great question. And I would say from a collaboration standpoint, we really look to continue these efforts and building on from what we learned from step 1 and expanding to potentially include additional sponsors. We're learning more through these efforts. And as mentioned earlier, we know we can't do it alone. So it really takes efforts like this with lots of stakeholders. And and I would say owning this collectively to move the needle. That being said, circling back to what we noted early at Genentech efforts, we were focused on earlier stage disease, and I think this is a wonderful opportunity to hone in on this as a selection tool to customize care for cancer patients. And I would finally add, I think from a patient journey perspective, as it pertains to priorities moving forward, having been in the clinic for 25 years, it's always important to appreciate cancer care from a patient-centric standpoint and ask ourselves in industry how we can better understand the impact of our efforts on their lives.
Thank you. Thank you. Other thoughts on treatments that we might want to prioritize, or even clinical settings we want to prioritize in step 2?
Well, certainly, you want the patient to be on the most effective treatment for the longest period of time, but you don't want to keep them on a treatment that could have toxicity if they're not benefiting. So I think the ability now to switch up treatments, in lung cancer, I'll just focus on that because that was this project, but this should be brought out to all different cancer types. But, resistance develops so quickly in some cases. So understanding when the patient becomes resistant and when to switch or add another agent, this could be a huge way to do this, because not only will we know what that patient has increases in ctDNA, but we'll also know what's happening mutation-wise. So I think as we move, I agree with David, to the earliest stages of disease. We're doing great in the metastatic setting, but it's at the earliest stages, post-surgery, post-radiation, that we can even have more benefit. This is going to be a huge advance, and it's going to help patients, and we'll be able to use it to help patients as we incorporate it into research studies.
Very good. Thank you. Other thoughts?
Well, I think the 3 broad categories are, after immunotherapy, would be targeted therapy. And is the behavior the same? Is the timing the same with the established targeted therapies? And that could lay the groundwork for new targeted therapy development, which, as we know, is ongoing. And then certainly chemotherapy would be interesting. I worry that kinetics are going to be different with chemotherapy. There are, of course, emerging chemotherapy tools, precision chemotherapies, antibody drug conjugates. And so can we learn from our basis of existing tools to lay the groundwork for how we would use this device and this technology for the next generation of tools that are emerging?
Very good. I'm going to change gears just a little bit, and I wonder if Dr. Julia Beaver is still available to us and if she might be able to join. Thank you. There you are, Julia. Welcome. We've gotten a question around surrogacy, and related to that is a question about how does ctDNA compare to, say, RECIST in terms of being associated with survival. So maybe just some general thoughts from you first around what should we all consider as a community in our roadmap that might describe different types of regulatory actions. And then we'll see if any of the other panelists want to chime in.
Sure, thanks. So for ctDNA to be used as a novel endpoint, it's important to have biologic plausibility, which there's already evidence supporting. Also studies of prognostic value of ctDNA with respect to the clinical outcome, and that seems to be what we're generating now. And in particular, I think it would be helpful to further longitudinally examine the timing, as was mentioned by a number of panelists, examine the timing of the ctDNA assay with respect to outcome, as well as better understand ctDNA across diseases and treatment classes. And ultimately, the strongest evidence would come from prospective randomized clinical trials or a meta-analysis of similarly conducted clinical trials that demonstrate, for a context of use, that ctDNA is predictive of the treatment effect and long-term outcome. I think also you touched on comparing to response, and so it's also going to be important in the metastatic setting to differentiate ctDNA from objective response rate, or show how it could be complementary to, since although response rate is commonly used to support drug approvals and is relevant for clinical decision-making, it's not always been shown to track specifically with long-term outcomes. So if ctDNA could improve upon this or add to ORR response rate in some way, it could prove very useful in regulatory decision-making.
Very good, thank you. Let's go to Jeff, and then I see Roy also wants to chime in. So Jeff first.
I want to tag team on something I think I heard Dr. Beaver say, which is that, there's surrogacy, which is this the endpoint that will get a drug approved? But before that, there are all sorts of ways that we can use endpoints earlier than surrogacy to anticipate response, to help us know if a drug is at the right dose potentially, to help with correlative endpoints. And so, to my mind, the first thing studies need to do is collect this specimen. If you're not collecting plasma, you're already behind. But beyond that, it's not enough to collect the specimen and save it for later, right? There's stuff to be learned today, we're finding from this data. And so let's analyze those specimens to accelerate and advance our drug development towards some of those opportunities that Dr. Beaver was highlighting.
Good, thank you, Jeff. Roy.
Yeah, I agree completely. So the biggest problem, we have in the metastatic setting is with immunotherapy, as amazing as it is, still 80% of patients progress, maybe even more. And then we're looking for new combos in that setting. And I gotta be honest, sitting there looking with a radiologist to measure films, it's pretty hard. How do you look, using RECIST criteria? Pseudoprogression, it's not that common, but it's real. Pneumonitis, it's real. There are so many other factors. So to have an objective measure like this that we could use to say someone is progressing, and then we define progression, and then we see response, this is going to be the way that some of these IO immuno-oncology combos are really, I think, going to move forward. So I think it can be used immediately and then validated in that type of setting.
Very good, thank you. David.
Yeah, I would add, just building on what Roy said and Jeff, very eloquent answers, is that this is important for so many different reasons here. And if we can better identify patients who might benefit most from, to Roy's point, adjuvant therapy after chemoradiation as an example, this really could influence how we design our trials moving forward. So one could imagine we might be able to limit the number of patients enrolled in these trials who might not benefit from the treatment. So I would say as a surrogate endpoint, ctDNA is a very exciting possibility. But again, to everybody's point, we recognize we'll need more data to support this over time and look forward to continued collaborations to try to accelerate this as a potential surrogate endpoint.
Very good. So I'm going to pause now. I want to get us more focused on step 2, but I'm noticing a lot of questions that have to do with the specifics of the analysis. So I'm going to go ahead and try and respond to a number of them. So first, the work has not yet been published, but it will be, and I'm sure that the team at Friends of Cancer Research will let everyone know when that's available, and it will have quite a few of the details you're yearning to hear more about. Second, each cohort that was presented, actually each sponsor had a different ctDNA assay from the other. So just know that, and we were still able to harmonize in a meaningful way. And last, there's a question about the specificity of the variants, and it's an overall variant load as opposed to a matched pair variant change, at least in the step 1 in the retrospective step. And on that note, yes, you had to have a baseline. The results that you've seen are all based on change from baseline. So, and I think there were just a handful of patients for which we weren't able to have a baseline within the window of 14 days prior to the initiation of treatment. Okay, so that said, I want to get back to what might be the most ambitious set of goals we would have for ourselves in step 2. And Roy, I'd like to come back to you around, is there something that we could specifically look at in step 2 that would then have the most impact in terms of clinical care in the future?
Right. Well, I think, and what I've already said, let's start to use it, to correlate with response and understand, in certain areas, I think just knowing that we had a defined response to a combination in a patient who's already progressing would be a great advance and would be helpful. And you would assume that that could help with patients' symptoms and quality of life. So I would say just to use it, in patients who are receiving these drugs to see if it goes down or not, and how that correlates with the radiologic response. I think that could be done right away. I know that sitting in the clinic, I often am trying to decide, do I keep the immune therapy going or not? In someone who has either a stable disease or maybe a little bit of progression, within that window of stable disease. And I think that one could immediately start to use this type of technique. Now that we know that there is some harmonization, there are a number of techniques out there. So we know we can get one of these assays and use that to help clinical care. So immediately helping patients with cancer to benefit. So I think that's something that I think can be done almost immediately.
And related to this, imagining things that would affect clinical care, there's a question that's come up around, should we look to centralize how the ctDNA assays are being conducted and anything to do with the assay to assay variability that we should look to understand, so that when you're looking at results, you kind of know where you stand.
I'm happy to take that. We've done some of that work in my prior time at Dana-Farber. It requires benchmarking, right? It requires having, two paired specimens that are studied on two parallel assays. That's hard to do with plasma because it's hard to get a lot of plasma and study a lot of assays in parallel, but it can be done. There are some other efforts to develop standards by which you can benchmark different assays in parallel. So I think that would be a really helpful solution. If we had quantitative standards that any assay could benchmark against and measure as tumor content goes up and down, that would really advance us towards something everyone can use. But it's true, all these assays have a slightly different way of measuring tumor content, and we do need to be intentional about that as we figure out which assays are well suited for this application.
And Antje, coming back to you in terms of the statistical aspects of having multiple assays, if we have the luxury of a prospective plan, is there any recommendation you would make to us as a group?
I mean, some sort of a reproducibility study would be fabulous to determine whether assays are equivalent and comparable, where, just like Jeff was saying, if we could take ctDNA samples and send them to different assay companies and compare the results, that would be the gold standard. That would be the perfect world, right?
Yeah. Julia, there's another question for you. When we, and I think it's a question for all of us actually, when we talk about this prospective phase, do we mean that we prospectively design around ctDNA, or that we're more intentional around including it and the analyses we would then undertake?
Yes, so you would want to be intentional about designing around ctDNA, using that in a standard manner. And you could have within your statistical plan, a plan to correlate that with outcomes.
Okay, so I think that's a clear answer. There's also a lot of questions coming through, and this might be for, oh, did you want to add?
If you don't mind, I'd like to add to what Julie just said. I think it'd be fabulous, and we said that before, if we could have longitudinal data with pre-specified timeframes. If we could agree on some pre-specified windows and frequencies of samples being collected in a study, that would be
well,
gold standard.
We must have been working a very long time together, because you read my mind. I was going to go to the panel and say, okay, what time points, if we were to get really granular, what time points do we want to see? And I'm wondering, Jeff is coming off mute. I'm
happy to make an ask. I mean, again, I've been designing these trials, and I think about cycle 1, day 1, a really clean baseline specimen that's not a week or two old, that is really representative of the day you started treatment. That first time point, 2 or 3 weeks in, depending upon patient care, when they first come back in, that's the earliest time you could see it. And then probably I would say the time of the scan, which is maybe 6 or 8 weeks. I see those as some of the key 3 time points to convince you that most of the signal happens immediately. There's no additional signal you're missing between the first time point and the scan. If you want to collect a resistance specimen, that's always great, but it doesn't really apply necessarily to this response analysis.
Very good. Okay, there's some wonderful questions coming around what kind of partners and collaborators are we looking for in step 2. I would say absolutely get in touch with the team at Friends of Cancer Research, but you're getting a sense from the panel discussion that we're really looking for those of you who are running trials, who want to provide data, individual patient level data, and who, as Julia has mentioned, are interested in being purposeful and pre-specifying how this would be tying in together. So being part of the thinking process alongside the rest of the team here. I know that there are more questions, but time is somewhat short. And so what I'd like to do now is maybe close out the panel and hand it over to Jeff.
Thanks.
Thank you all.
Great. Thanks so much, Nevine. And thanks for everyone for joining the meeting today, and to all of our participants, particularly our collaborators who have really driven the setup and the execution of this terrific project. It's really been a true collaboration across all sectors, and we really thank all of you. I hope that through today's results presentation, you've seen the role that ctMoniTR can play in the advancement of a potential tool to accelerate drug development, which may help provide safe and effective new treatments to patients. In response to some of the questions, a recording of this presentation and the subsequent discussion will be made available in the next couple of days. And as a next step, we'll be following up with numerous interested parties to explore how we can expand these efforts into step 2, including looking into different treatment settings and drug classes. So please consider joining us in this effort. Also, please keep an eye out for our next virtual meeting, which will be coming in September. It will include the results presentation from our Real World Evidence Pilot Project 2.0. Until then, we thank you for your continued support of Friends, and take care.